Hello, everyone, and welcome to Indoor Biotechnologies' latest webinar on the molecular approach to allergy diagnostics, innovative product and services, where we're going to be providing an in-depth discussion of InBio's unique and exclusive tools for allergy diagnostics. We really appreciate everyone taking time out of their busy schedules to join us today. We've got a very well-attended webinar of approximately 185 or so registrants from people all around the world. So we're excited for this opportunity and appreciative as well. Today's webinar has got a great lineup of speakers. We'll be starting with Dr. Sabina Wunschmann, VP of Allergen Manufacturing R&D. Sabina will be speaking specifically to the purified allergens, mainly the manufacturing and purification process, as well as our stringent quality control measures. Next, we'll have Dr. Martin Chapman. Martin is President and CEO of Indoor Biotechnologies, formerly a professor of medicine and microbiology at UVA, and also a fellow at the American Academy of Allergy, Asthma, and Immunology. Martin was also the first ever recipient of the prestigious FARF Award, which is awarded to young investigators who have made outstanding contributions to the field of allergy. Martin is going to be discussing a new and exclusive product to Indoor Biotechnologies, which is Human IgE monoclonal antibodies. Martin will be providing a historical look at this technology as well as the impact. Followed by Martin, we'll have Dr. Crystal Richardson, postdoc scientist here at InBio. Crystal will be diving more into the Human IgE monoclonal antibodies, discussing the development of these antibodies, as well as the data and potential applications. Finally, we'll have Dr. Ana Plamez. She will be discussing her research in human IgE epitope mapping and how the structural basis of allergenic epitopes can be identified using IgE monoclonal antibodies. Ana is also a fellow at the American Academy of Allergy, Asthma, and Immunology, and also a recipient of the FARF Award. And I'll be your moderator, Beatty Sturgill, Business Development Manager here at InBio. So to provide some structure to today's webinar, again, we're going to start with the purified allergen proteins, that manufacturing process, quality control measures, and applications. We'll then move into the new product, Human IgE monoclonal antibodies, where we'll discuss the history, impact, applications, and the human IgE epitope mapping research. We'll conclude the webinar with a Q&A section. Please submit any questions that you have using the Q&A feature found in the Zoom toolbar. You can click on the Q&A button. You can post questions associated with your name or anonymously at any time during the webinar, and we'll go through as many as we can at the end of our presentations. With that, I think we're ready to get started here, and I'll turn it over to Sabina.
Yeah, thank you, Beatty, for the introduction. So we'll jump right in and be talking about allergen manufacturing at Indoor Biotechnologies, from extract to purified allergens. Next slide. So because the purified allergens are the basis of molecular allergy diagnostics, I thought I would give a brief overview of this application for purified allergens. So molecular allergy diagnostics focuses on the allergens at the molecular level. Instead of measuring IgE reactivity to the sum of all allergens in a source such as peanut, molecular allergy diagnostics measures IgE reactivity to the individual allergens, in this case to the individual specific peanut allergens such as RH1, RH2, or RH3. Next slide. Measuring the IgE reactivity to individual allergens will help establish a patient sensitization profile that will allow to distinguish between specific allergens shown here in red that are the primary sensitizer versus cross-reactive allergens shown here in blue. Next slide, please. So besides the information on cross-reactivity, molecular allergy diagnostics also allows some insight into the risk assessment for each patient. So allergens belong to different protein families, which will cause symptoms of varying severity. Measuring the IgE to individual allergens and knowing the associated families and protein families will allow to do a molecular profiling for each patient to establish if the patient has a low or high risk for severe reactions. So for example, if a patient is sensitized to allergens of the profilin family that are very labile proteins, the risk associated with a severe reaction are very low. Whereas if a patient is sensitized to storage type proteins that are very resistant to heat and digestion, the risk for developing severe allergic symptoms are much higher. So now that we know a bit background on the molecular allergology and this, which is an important application for purified allergens, I will move over to talk about some technical details on allergen manufacturing here at Indoor Biotechnologies. Next slide. So all purified allergens are manufactured at our Charlottesville, Virginia headquarters location using an in-house, using an ISO certified quality management system using ISO standard 9001-2015. Next slide. Indoor manufactures over 100 different purified allergens that are either natural or recombinant. So the natural allergens are purified from natural source materials such as peanut flour or dust mite culture, whereas the recombinant allergens are expressed either on Pichia pastoris or on E. coli. Both the recombinant or the natural allergens can also be expressed and manufactured as low-tox allergens that have a very low endotoxin level below 0.03 EUs per microgram of protein. These are ideal for studies for human or marine cellular studies. Our current repertoire includes allergens from foods, dust mite, cockroach, animal, pollen, venom, and mold. Again, we are about over 100 allergens at the moment. Next slide. The considerations for manufacturing of natural allergens include that we need to have a reliable supply and a reliable supplier. There's some inherent variability associated with using natural source materials, such as the pollen may be harvested at different times of the year. We're using dog hair to extract dog allergens that come from different dog breeds, etc. Some allergen materials, such as pollen, can be very costly. And then some source material may have already undergone some processing, such as peanut flour that has been, where the peanuts have been roasted already. Once we get the materials here at Indoor, we have to figure out the optimal extraction conditions and figure out temperature, pH, and best buffer conditions to have soluble allergens. One advantage of using natural allergens is that they represent the natural form of the sensitizing allergen and the relevant mixture of isoforms as they occur in the natural allergen. And they also have the natural post-translational modifications. Purification, we have to figure that out. Usually we use HPLC, purifying by charge or by size exclusion, or we develop monoclonal antibodies to develop an ELISA assay, and then we can use those monoclonal antibodies on an affinity column to purify the allergen from the natural source material. Some of the allergens, especially the peanut allergens, need a lot of different depletion steps to get rid of co-purifying trace amounts of other allergens that we need to deplete. Next slide. So recombinant allergens have been shown to be very similar or equal to their natural counterparts. regarding the ability to bind IgE, to stimulate T cell responses, and to trigger an allergic response. So for recombinant allergens, we can express them as pure defined molecules, either expressed in E. coli or Pichia. They can be scaled up easily, and they can be produced under standardized conditions with a controlled quality. Since we have to decide on one amino acid sequence to express, we need to choose a specific isoallergen. Usually we orient ourselves at what has been published or what the abundance is of the isoallergens in the natural material. For purification, we tend to add an affinity tag to either the N or C terminus of the protein. We use histidine tags between 6 and 10 histidines. And then our first purification step is usually to purify recombinant allergen by IMAC, and then followed by size exclusion chromatography, as you can see here in the lower right quadrant, which is a typical size exclusion profile, and we select the blue colored protein. Also here in the upper right, you can see the model structure of a cannabis allergen, Canis3, and we think this allergen may gain some diagnostic relevance, as cannabis has been legalized in many US states, and is a lot of other states are following. So we think that there will be more exposure and more allergies to cannabis that need a diagnostic reagent to diagnose. Next. So we start the design of a new recombinant allergen by finding out what is the mature allergen sequence of the natural allergen. To do that, we use some predictive software that tells us about cleavage of the amino acids, that tells us about signal peptides and where the protein chain may be cleaved. So once we have that, as I mentioned, we need to decide on the isoform to express. Another consideration is to see what the glycosylation will look like of the expressed protein. That's particularly relevant for expressing in Pichia pastoris. On the lower right, you can see one of our predictive softwares that will predict 2 glycosylation sites for this protein that we tend to then silence, so we don't have hyperglycosylation of yeast-expressed proteins. And then another software that we use is to model the allergen before expression, to look at the structure and to look which, if the N terminus or the C terminus is the best place to add the purification tag, the histidine tag. In this case, you can see that the C terminus in red is rather floppy and probably prone to degradation and cleavage. So for this allergen, we would use the N terminus in blue to place the tag. Next. So every allergen is validated for purity using mass spec and SDS-PAGE, for IgE binding, and also for stability. Since I cannot show data for every allergen, I chose one example to go with here, and I chose recombinant ALT1, the major allergen of Alternaria, which is a 15 kilodalton allergen. And when we run it on SDS-PAGE, as you can see on the left-hand side under non-reducing conditions in lane A, the Pichia expressed allergen runs as a dimer about 30 kilodalton, whereas the E. coli expressed protein that we compare it to runs as a dimer and as a monomer. When we reduce the Pichia expressed protein, it drops down to just the monomer in the inset shown here. Drs. Max Cruz and Dr. Anna Pomez here from Indoor have also worked on the crystal structure of this allergen and have shown that in solution, the ALT1 also presents as a dimer in the shape of this beautiful butterfly that you can see here on the upper right. Next. So all allergens are validated by mass spectrometry, and Indoor has purchased a couple of years ago an in-house mass spectrometer, a Thermo Fisher Q-Exactive. And so we use a mass spec to during allergen development to make sure that we have the correct proteins. So we use it for protein identification, and then for purity assessments by relative abundance. Every allergen lot that we produce is QC'd and is not released until we have done the purity assessment by mass spec. And of course, during validation and development of new allergens, we use it as well. For the ALT1, you can see we have a good sequence coverage of the protein on the upper part of the slide. And then we have a purity by a relative abundance of over 99%. Next slide. So as I mentioned, every allergen is validated for IgE binding. And then the graph on the right side, you can see that the IgE binding between E. coli and Pichia-expressed recombinant ALT1 shows an excellent correlation. In this case, using a chimeric IgE ELISA. And then if we have available a monoclonal antibody ELISA for detection of the allergen, which we have for ALT1, we can also see the reactivity of recombinant ALT1 by ELISA assay. And you can see both the recombinant E. coli and Pichia-expressed ALT1 show an excellent, very comparable immunoreactivity by ELISA assay. Next slide, please. So each allergen will enter a stability study that we do in real time over a period of about 5 years now, where we measure at different time points the protein concentration by OD280 in this case. If we have an ELISA available to measure the allergen, we will run the ELISA, and we do an SDS-PAGE to see if there are any signs of protein degradation. And as you can see here, we're not quite at the 5 years yet, but the ALT1 looks really good. After 48 months, we don't see any signs of degradation compared to the starting time. And both the protein concentration and the ELISA show a very solid reactivity over time. If there are any concerns, we will shorten the shelf life and adjust our manufacturing accordingly. So now that I have shown the technical aspects, I want to finish up with new allergens that are coming out soon and additional applications besides molecular allergy diagnostics. Next slide, Beatty. So the allergens that we are working on currently and that will be released soon contain additional tree nut allergens from Brazil nuts, pecan, pine nut, and almond. And we also have strawberry and egg allergens, as well as some lipocalins from dog and uram-1 from dust mite. So those will be released soon, and you should see them on our website and our upcoming newsletters. Next slide. So Indoor has been consistently working on expanding the range of the purified allergens over the past few years to serve the molecular allergy diagnostic market. And we are pleased to work with several manufacturers of in vitro diagnostic products to serve as a supplier of purified allergens. So besides this important application, there are other applications, of course, for purified allergens, which include in vitro research, doing mouse models of asthma, T-cell studies, food allergy research, etc. They can serve as standards in immunotherapy, drug development, as well as for allergen standardization. Next slide. So to summarize, Indoor is producing over 100 allergens as well as viral proteins from rhinovirus and SARS-CoV-2 proteins in-house under ISO 9001:2015 certified quality management system. And all of our allergens are protein ID'd and the purity verified by mass spectrometry before we release them. And they're also validated for IgE antibody binding. And with that, I will close and turn back over to Beatty.
Thanks, Sabina, for that great presentation there. Just a reminder to everyone, if you have questions for Sabina on our purified allergen proteins, please type them using the Q&A feature in the toolbar, and you can post them there. But now we're going to change tracks a little bit and speak to, again, one of our newest products, which is the Human IgE monoclonal antibodies. And we'll start with Martin, who's going to give us a historical overview of this technology. Martin?
Hey, well, thanks, Beatty, and welcome to all our friends, colleagues, and customers from all over the world who've joined us on this webinar. Beatty's only given me 5 minutes, so this is a brief 5-minute history of IgE, and also its relevance to what we're going to talk about today. The allergy field in the 1960s was transformed by Ishizaka's discovery of IgE as a new class of immunoglobulin. Previously, reagenic or skin sensitizing antibody, many people thought that that was IgA antibody. Through a rather thorough and painstaking series of experiments, Ishizaka convincingly showed that IgA antibodies did not have reagenic activity, and that the Prausnitz-Kussner skin testing correlated with a new fractions, a new size of immunoglobulin, which he called IgE. Now, serendipitously, and really rather remarkably, at the same time, 2 IgE myeloma proteins were discovered, IgE-ND in Sweden and PS at Dartmouth College in the US, I'm sorry. Now, just bear in mind that there have only been about a dozen IgE myeloma proteins ever described. And so the idea that there were 2 of these appearing at the same time as this discovery of IgE was really remarkable. It allowed a lot of structural studies. The IgE-ND was discovered by Hans Bennick and Gunnar Johansson at the Karolinska Institute. And that really set the stage for collaborations between these 2 groups, the group in Sweden, Johansson, and also Ishizaka's group, to define IgE antibodies. What came out of those studies was the absolute amount of IgE in allergic serum was very low, and that has had implications for the field ever since then, 50 years ago. Beatty mentioned the FADIA Pharmacia Allergy Research Foundation Award. I have a personal history of that. Gunnar Johansson in 1987 recommended the formation of this award to celebrate the 20th anniversary of IgE. And I met him at the Allergy Congress in 1987 at the American Allergy meeting, and he informed me that I'd won, which was great. And the prize was actually presented that year by Dr. Ishizaka, which was really, as a young scientist, quite incredible. Then things, I guess, went full circle because I had the tremendous honor in 2016 to actually review what was probably Dr. Ishizaka's final paper, which I encourage you all to read because it's just a tremendous description of how that IgE was actually discovered. Next slide. Subsequently, Gunnar Johansson and Leif Vida went on to describe the radioallergosorbent test for measuring IgE antibodies, and that became pivotal for serological diagnostic methods. The World Health Organization acknowledged IgE as a new class of immunoglobulin and developed a standard. At that time, people were measuring IgE antibodies both in terms of the WHO standard and the PS myeloma serum. And so Bazaral and Hamburger did a study where they compared the 2, and they showed that, and they came up with this thing that 1 international unit of IgE was 2.4 nanograms, and that has been the definitive definition of a unit since 1972. So moving on, and this is my final slide, we've been very fortunate to collaborate over the past 3 years with Dr. Scott Smith at Vanderbilt University Medical Center. Scott really did the thing that nobody else had done. He tried to make human IgE antibodies in a fairly conventional route from IgE from allergic patients and to identify antibody-secreting B cells and then fuse them with a myeloma cell line. When Scott was doing this, he initially worked with Aspergillus. And then he needed more allergens because to identify these antibodies. So he'd been collaborating with Anna Pomaeus, who's been looking at the allergen specificity of these antibodies, doing some epitope mapping and some really super localization studies of IgE to contact residues. As a result of this collaboration, Indoor Biotechnologies signed an exclusive licensing agreement with Vanderbilt University Medical Center, and that licensing agreement covers use of human IgE antibodies for research and diagnostic applications. So we're excited about this new IgE monoclonal antibody technology. We think it has lots of potential, and I'm going to hand over now to Dr. Crystal Richardson, who is heading up our IgE antibody manufacturing and production process. Crystal.
Thank you, Martin. As Martin said, I'm Dr. Crystal Richardson from Indoor Biotechnologies, where I serve as the project leader for the manufacturing of the human monoclonal IgE antibodies. And today it's my pleasure to tell you about the science and technology behind these antibodies. Next slide. There are several advantages to using human IgE monoclonal antibodies for research and diagnostic testing. So typically, these experiments would be done with the sera from allergic patients. However, the IgE antibodies in human sera are polyclonal and found at very low concentrations in the nanogram per microgram amounts. Our human IgE antibodies are monoclonal and can be isolated at very high concentrations in the milligram range. Additionally, unlike recombinant antibodies, human IgE antibodies are obtained from B cells of allergic patients, which means they contain the natural pairing of heavy and light chains. This also means that human IgE antibodies may be used as an alternative to human sera. Next slide. In order to develop this technology, we collaborated with Dr. Scott Smith from Vanderbilt University. The methods are as follows. PBMCs are isolated from blood cells of allergic patients. Second, the B cells are expanded in culture. Third, IgE secreting B cells were identified. Fourth, these B cells were fused with a myeloma partner to immortalize the cell line. Fifth, the cells were grown and expanded, selecting specifically for high IgE secreting cell lines. And then lastly, the cell supernatants were collected and purified using affinity chromatography. Next slide. This technology has allowed us to create a panel of 15 human IgE monoclonal antibodies to inhaled and food allergens shown here. So to validate these antibodies as IgE, we used immunocap to measure the total levels of IgE. What we found was that our human IgE antibodies can be purified at incredibly high levels, all the way up to 2 million units per mil. Next slide. The purity was established by SDS-PAGE that has been silver stained. Shown here are 6 representative examples of IgE antibodies to inhaled or food allergens. In all cases, the heavy and light chains are seen, and the purity was established as greater than 95%. Next slide. Additionally, reactivity was determined by ELISA. Shown here are the same 6 antibodies tested for reactivity to their respective allergens. In all cases, the curves are sigmoidal, with the limits of detection being in the nanogram per mil range. For the next slide, I'll focus on the last 2 antibodies, 1E7 to egg allergen GALD4 and 16A8 to peanut allergen ARH2. Next slide. So allergen specificity can be established by immunoblot. First, I'll discuss 1E7 to GALD4, and then next, I'll talk about 16A8 to ARH2. For both of these, a Kamasi stain was done, so you could see the total protein that was loaded onto the gel. So focusing first on the left, 1E7 to GALD4, what we saw when we did the blot was that GALD4, which runs at just under 15 kilodaltons, that band was very apparent when we probed for the purified GALD4. Additionally, looking at this non-reduced lane, we see that there's a higher molecular weight band that's about double the size. This suggests that it could be a dimer of GALD4 that these antibodies reacting to. Looking at the last 2 lanes, the total egg extract, what we see in the non-reducing lane is several higher molecular weight bands, one that looks to be about the size of the dimer, but then some others that could be some multimer complexes, or perhaps Gal-D4 is actually binding to other proteins in the egg extract. In both cases, when the sample was reduced, these higher molecular weight bands seemed to disappear, and only the band that corresponds to Gal-D4 was apparent. Looking on the right-hand side, where we probed the blot with 16A8 to ARH2, we see a doublet that corresponds to the size of ARH2 between 15 and 20 kilodaltons. That's very apparent in the purified lanes. And then looking at the total peanut flour protein extract, what we see again are several higher molecular weight bands that may be that ARH2 binding to other proteins. However, when the samples were reduced, the most apparent bands do correspond to ARH2. Next slide. For all antibodies, rigorous quality control experiments were done. The total IgE levels were standardized by ImmunoCAP. The antibodies were found to be greater than 95% pure by SDS-PAGE, and the antibodies reacted specifically with their target allergens by ELISA. There are several applications for these human IgE antibodies, including Allergy Diagnostics, using the antibodies as IgE standards or to identify allergens, as well as several biological assays, including mast cell and basophil activation studies, histamine release, and the antibodies can be used as animal models or used in animal models for anaphylaxis. Additionally, Dr. Anna Pomes will talk about epitope mapping with these antibodies. Next slide. And I'm also excited to tell you that we'll be expanding our panel, adding 3 new additional antibodies. So we'll have 18 total human IgE antibodies. We have this antibody 1C14 to DERP1, another one 2F10 to DERP2, and then also 1B7 to the cat allergen Feld-1. And then shown below are our quality control analysis. In all cases, the antibodies were pure at greater than 95% on SDS-PAGE, and then they are reactive to their specific allergens by ELISA. Thank you.
Great. Thank you, Crystal, for that presentation. And next we can move to Anna, who will talk about the human IgE epitope mapping.
Good morning. My name is Anna Pomes, and I am the Director of Basic Research. Here at Indoor, we have a research program that aims to investigate antigenic determinants recognized by human IgE for the design of immunotherapy. So this research program is funded by the National Institute of Allergy and Infectious Diseases, the NIH, through an R1 award for which I am the contact PI. Next, please. So the rationale for doing those studies is the limited knowledge we have nowadays of the epitopes associated with the human IgE repertoire. So one of the reasons for this is that IgE, as you know, is polyclonal in low amounts in the blood, and also the efficiency of the B cells that produce IgE is really low in blood. So the aim is to identify and analyze epitopes for IgE for the subsequent design of hypoallergens for immunotherapy. Next, please. So over the years, we have addressed this aim by determining the structures of allergens in complex with antibodies. And we started with the cockroach allergen BLAG2. You can see here on the left, that's a superposition of 2 of those complexes, showing BLAG2 in the middle, and then 2 of those antibodies binding to the opposite lobes of BLAG2. So those studies originally were done with Dr. Alex Flodawer's group at the National Cancer Institute. And later on, we have worked a lot with the group 1, mite allergens. We've done that in collaboration with Dr. Max Cruz from the University of South Carolina. And we have studied DERP1, DERP2, and here on the right, you can see a superposition of 3 of those allergen antibody complexes with DERP1. So in all those cases, we're using murine IgG monoclonal antibodies that inhibit IgE. And those antibodies are used as surrogate of IgE. Next, please. So the most complex that we determined was with DERP2. again, an IgG mutant monoclonal antibody FAB in complex with LP2. And on the left, you can see a diagram of the secondary structure of the heavy chain of this antibody. And you can see all those beta strands that form the immunoglobulin-like domain. And then the loops connecting those strands, 3 of them in color, contain the CDRs, or complementary determining regions of the antibody. Those form the paratope. So by solving the structure of the allergen with the antibody, we can see exactly where those CDRs are recognizing the epitope, and then select residues on that epitope that we can mutate and create mutants that no longer bind less the antibody. So in this particular study, the mutants of their P2 no longer bound in IgE construct that was obtained from a binatorial library by Dr. Mats Olin from Lund University in Sweden as a collaborator. Next, please. So with the advent of this human hybridoma technology developed by Dr. Scott Smith, we have been able to obtain a panel of 4 anti-DERP2 monoclonal antibodies and do mapping studies. So we did that, we did relative epitope mapping with immunoassays on the right, and then in collaboration with Dr. Jeff Miller from the NIHS, we performed a epitope mapping by NMR. So on the left, you can see the P2, and 3 of those IgE antibodies bind to overlapping areas at the bottom here on green. And then the other one is called 2F10, binds to an opposite side of the molecule, and you can see it here in purple where it binds. So we selected this 2F10 just to further analyze it by X-ray crystallography. Next slide, please. So this is on the left, the structure of the P2 complex with an FAB of this antibody. And we're very excited about this because this is the first structure we have ever solved where the IgE contains the right pairing of the heavy and light antibody as it occurs in vivo. So the resolution was high at 2.2 angstrom resolution. So that allowed us to identify very clearly where the epitope is. And again, we did a site-directed mutagenesis analysis and created several mutants, some of which were able to bind IgE, but at 1,000 times less. So this is the kind of aim, of goals that we want to achieve in order to eventually get hypoallergens for immunotherapy. So next, finally, just to summarize, mapping human IgE antibody epitopes allows the identification of the residues that are important for antibody recognition. So this epitope analysis will facilitate the design of hypoallergens for immunotherapy and also contribute to understand the IgE antibody repertoire. And also these antibodies will have other applications such as therapy in a similar way as Regeneron has presented the anti-Feld-1 IgG antibodies. Those are our main collaborators in these current studies, and thanks for your attention. And I guess I will now hand it over to Beatty.
Great, thanks Anna, and thanks to all our other presenters for their great talks here. So all of these resources and others can be found on our website, InBio.com. That includes the purified allergens that Sabina spoke to. We have a comprehensive portfolio of natural, recombinant, low endotoxin, and biotinylated allergens. Crystal spoke to the development of the human IgE monoclonal antibodies. Currently, we have a panel of 15 available to both inhaled and food allergens. And as Crystal mentioned, we're continuously adding to that panel through the exclusive licensing agreement with Vanderbilt University. We also have a portfolio of monoclonal and polyclonal antibodies. These are developed against our purified allergens. And we have a wide array of immunoassay kits available. That includes our ELISA 2.0 kits, which are pre-coated plates that contain all the reagents necessary to run the assay and have results in 2 hours, as well as our Maria multiplex array, which is run on the Luminex technology. The Maria provides for simultaneous detection and quantification of multiple allergens from a single sample. Sabina briefly mentioned as part of our QC process for purified allergens that we assess the allergens for purity using mass spec. We also have standard mass spec services available to customers. That includes protein identification from a gel band or liquid, protein profiling of complex samples, or intact mass. Of course, we also offer custom biotinylation services of our purified allergens and contract research opportunities, which could be custom purifications, custom extractions, or immunoassay developments. Some of you may be aware that we have signed a distribution agreement with Millipore Sigma. We're very excited for this opportunity, as Millipore Sigma will be a global distributor of InBio's purified food allergens. Again, they're going to distribute our comprehensive range of natural and recombinant food allergens as analytical standards. The transition of these products should be complete in the coming months, and we'll be sure to share more details on that as they come true. And with that, that concludes our presentation portion of the webinar, and we can move into the Q&A section. Again, just a reminder, you can post your questions using the Q&A button on the Zoom toolbar. I'm going to stop sharing my screen now and pull up. Looks like we've got some questions coming in. We'll get through as many of these as we can. Sabina, I have one here for you. Are all of your recombinant allergens, do they all have his tags?
Yeah, so that's a good question. In fact, that not all of them do. A lot of our dust mite, cockroach, and cat allergens, recombinant allergens, do not have a his tag. Our new allergens, so particularly the food allergens, all do have a his tag. That's usually 6 histidines. Yeah.
Okay. Great. Next one here, are you ISO 17034 accredited? I believe that is the analytical standard accreditation. I'm not quite sure right offhand. While we're ISO 9001 certified for our allergens, we're not ISO 17034 accredited. But again, as I mentioned, we do have the distribution agreement in place with Millipore Sigma, where our food allergen proteins will be distributed as analytical standards on their website.
Betty, if I could just comment on that. We are also ISO certified for ISO 17025 for Maria testing and immunoassays. We are looking at ISO 17034 because it's usually an accreditation for standards. Indoor Biotechnologies has produced actually 4 internal reference standards for the Group 1 and Group 2 dust mite allergens, and we are looking to produce other standards as well. So I think that we are really, I also believe there have been some changes in the ISO 17034, which we'll be getting Brian Smith to look at. But this is definitely, now that we've gotten these other 2 ISO standards together, we're certainly looking at ISO 17034 as a possibility moving forward.
Great, thanks, Martin. Let's see, Sabina, I think this one will be for you as well. How is the consistency and critical QC parameters between the different lots of purified allergens determined before it is released as the reference standards?
So for the purified allergens, we, as I mentioned, we manufacture under ISO 9001:2015, which means we're using SOPs to consistently manufacture under the same conditions and using this exact same steps. We, for recombinant allergens, we use the exact same clone. For natural source material, we buy that in bulk. We test it before we use it. So there's a lot of consistency there. Our mass spec measurement before we release a lot needs to fall above 95% by relative abundance. And in fact, most of our purified allergens are at 98% or higher. The concentration is always adjusted to fall between 1 and 2 milligrams per ml by OD280 usually, or BCA. We look for consistent banding by SDS-PAGE using silver stain, and we look for consistency if we have an ELISA available for similar immunoreactivity by ELISA. And then our endotoxin levels are checked for every allergen, and for the low-tox allergens, it is adjusted to fall below 0.03 EU per microgram of protein.
Great.
Yeah.
Thank you. This will be for Human IgE. Do you have any examples of research applications of IgE and mast cell degranulation?
Yeah, so that's a good question. So we have not done the biological assays yet. We're actually working in collaboration with Dr. Ronald van Ree in Amsterdam to do, we're actually going to start with some basophil activation studies. But yeah, right now we are looking into some research as far as the epitope mapping goes, which will help us to identify which antibodies would be good for these types of cross-linking experiments. Yeah.
Thanks, Crystal. Another one here. Could the monoclonal IgE antibody be used for sequential IgE epitopes mapping?
Yes. So we actually.
This one is for me. Oh
yeah, you can go, Anna.
That's all right. In fact, yes, there is no reason why they should not be finding linear epitopes if that was the epitope that originated IgE. I just want to remind that those antibodies have been isolated by a hybridoma technology from B cells. So they are from the patient. So if the patient developed IgE against a conformational epitope, they obviously will bind better to that epitope. But if they were generated against a linear epitope, maybe some food allergens, that would be more common, then those IgEs will bind linear epitopes, and they can be used for linear epitope mapping. So some may not bind. Depends how they were generated by the patient, by the subject. Have anything to add?
Great.
Yeah, I guess I would say that the likelihood for inhaled allergens that they develop against linear epitopes, I would say, is small. But you know, I wait to be proved wrong on that. We'll see how that goes ahead. Yeah.
Sabina, I believe this question will be for you. Have you looked for O-linked glycosylation in Pichia-produced allergens?
No, actually, we have not. I believe the software we are using is just predicting N-linked glycosylations, so I will look into that. Generally, we do not see excessive hyperglycosylation in the Pichia, just focusing on the silencing the N-linked glycosylation motifs.
Okay. Another one for you, Sabina. How do you test that recombinant allergens maintain the same structure and functionality of natural allergens?
So we don't have for every allergen the natural counterpart or the recombinant counterpart, but for the few that we do, it was very comparable. So Betty, do you have time? Maybe we can pull up, we have a FeLD1 example of comparing IgE reactivity between natural and recombinant FeLD1. That looks like an excellent correlation. Yes, thank you, Betty. Here on the right, where we measured IgE reactivity comparing recombinant FeLD1 with natural using the chimeric IgE ELISA, and then using a FeLD1 specific ELISA assay, we also saw superimposed curves comparing the reactivity of natural Feld-1 to recombinant Feld-1 expressed in PKR or in E. coli. So we have a similar example for RH2, where we have a recombinant version and a natural version, and the IgE binding and ELISA reactivity is very comparable and correlates well. So for the few examples that we have, both forms, we see very good correlations.
Yeah, but Beatty, I guess I would also say that for some allergens, we actually only have the recombinant. And for example, if you look at blomia allergens, there are very few natural blomia tropicalis dust mite allergens out there. Most of them are recombinant, and I'm sure there are probably other ones that I can't think of at the top of my head now. So it really is a question of, if we have recombinant, if we have natural, we can do the testing. We try to get together panels of IgE antibodies so that we can do that. We can obviously do that now with the IgE monoclonals as well. But otherwise, we're really looking at the reactivity of either the recombinant or the natural in isolation. So that's at a practical level how we address that.
Mm-hmm. Great. I have another question here. Have you found that homologs have similar epitopes?
Okay, I'll get that one.
Mm-hmm.
Yes, in fact, they do. We have an antibody that's cross-reactive between DERP1 and DERF1. The name is 4C1, and we have the structure of both, DERP1 with that and DERF1 with this antibody, and they are recognizing the same epitope. There might be a few amino acids that are different, but it's very nice when we saw that they recognize the same site.
Mm-hmm.
So yes.
Thanks, Anna. Let's see, I have one here. Thoughts on the origin of IgE and evolution regarding to eliminate parasites. Have sources of IgE, i.e. B cells, ever been isolated from patients with parasite infections? Do they behave differently?
Yeah, I'll get that one. I know for sure that there's been, Scott Smith has been isolating a lot of IgE antibodies from parasites. And, you know, there is a lot of thoughts about the origin of IgEs as a way to defend against parasites. So, but what is interesting, I just heard him presenting those antibodies in the Swineflu conference last week. And basically, they are identifying proteins that are not homologous to the allergens. And that's interesting because, for example, we have an allergen that's a glutathione transferase from Ascaris. And you would think that that would be very commonly happening, that those helminths have homologs to allergens, but that's not the case. In fact, he identified the specificity of those IgE monoclonal antibodies, and they were against proteins that we have no idea that they have no homologs to allergens. Mm-hmm. So yes, they have been isolated, they are different, and he is in the process now of getting deeper analysis of those IgEs.
Mm-hmm. Okay, thank you. I think this is just a comment back to a previous question. O-linked glycosylations in Pichia do not hyperglycosylate, but can still be a problem that will interfere with immunoassays.
Great, thank you.
Crystal, I have a question here for you. Why do the SDS-PAGE gels molecular weights of the human IgE antibodies differ from the allergens?
Yes, good question. So on the SDS-PAGE gels for the human IgEs, all you'll see is the heavy chain and then the light chain, whereas the allergens can be a range of different sizes, and you can see different banding patterns. But antibodies in general, specifically these IgEs, have a very stereotypic banding pattern. One band at the 75 kilodalton and one at the 25 kilodalton.
Mm-hmm, mm-hmm. Great.
I'd just like to address one of these other questions. I think there's a question about comparing the immunocap results on the IgE antibodies to specific IgE in the immunocap. That is something that we have in process. we initially decided to look at the total IgE as a good way of measuring each of these allergens. And initially, we decided to do the specific IgE measurements using our specific in-house ELISA, which seemed to work very well. But we are now looking in more detail for all of the antibodies at the behavior on the immunocap for specific IgE, both to the source extract and also to individual allergens where they're available. So there are purified allergens available on some immunocaps that we will be checking out. I think we need to, you know, we are still at the early stages of looking at the relationship between the monoclonal antibodies and polyclonal IgE responses. Obviously, the monoclonal antibodies are directed against single epitopes, and that could affect their it wouldn't affect their behavior in a total IgE assay, but it could well affect their behavior in other immunoassays, depending on how those allergens are coupled, whether or not the epitope is available or blocked. So I think there's a number of considerations that we need to look at to really go into this utility of the individual antibodies for calibration purposes and so on. So we're really at the early stages of that. And likewise, as Crystal mentioned, for the mast cell and basophil studies, what we're trying to do and are identifying are IgE antibodies to multiple epitopes, which we have for dust mite, which we have for Feld E1, which we have for some of the peanut allergens. And so, you know, if we can get those, if they will work to cross-link, then that's going to be, you know, it's a sort of missing piece of the puzzle at the moment, because we have good in vitro data on the IgE antibodies, but we'd like to get some more biological data just to confirm their significance. And unfortunately, these days, you can't do PK testing anymore, which is a bit of a shame, really. Anyway.
Great. Thanks, Martin. Looks like we've got time for one or two more questions here. Sabina, one for you. Should I use natural or recombinant allergens in a diagnostic application?
I think that both of, first of all, as we mentioned, we don't have necessarily, there isn't necessarily a choice for all allergens to either go with natural or recombinant. I think either works well, and as I mentioned when I was talking about the considerations for natural allergens, there may be some inherent variability, at least when it comes to the source material, that we wouldn't have with recombinant allergens. But if your particular way of using the allergen in diagnostics, if the his tag is a problem, then maybe a recombinant would not work. But we work with several manufacturers of allergen diagnostics that have devices and that are using our allergens, either biotinylated or otherwise, that they don't have a problem with a his tag being present. So I would say in short, both natural or recombinant are fine.
Yeah, thank you. And then the last question real quickly, it was mentioned viral proteins. What viral proteins does Indoor have available? We actually have a slide for this that I can put up real quick. We have recently developed some SARS-CoV-2 proteins, and this is listed here of the SARS-CoV-2 proteins that we have developed, such as the spike receptor binding domain, nucleocapsid RNA binding domain. We have low tox versions of those as well, and others in development. We can follow up with information when we send out the recording of the webinar regarding this.
Beatty, there's one last question I want to address, and that regards stability studies on the purified allergens. So I may have forgotten to mention that when I presented. So the stability studies are done at the recommended storage temperature at -20 degrees. We do not have room temperature or 4-degree data. We only have -20 degree stability data on the allergens.
Great, thanks, Sabina. Didn't see that one come in there.
That's
So,
okay.
let's see. Well, I think that's all we've got time for now. We're right at 12 o'clock. So do greatly appreciate everyone attending today. Again, it was a very well attended webinar with a lot of good questions here. So appreciate the time. Does anybody else have anything?
Thanks very much. Thank you.
Thank you.
All right, thanks.
Bye.